Welcome everyone to this SymbNET series. My name is Massimo Micone. I'm a PhD student in the Evolutionary Biology Lab here at IGC. We have two speakers. The first one is Jan van der Meer. I hope I pronounce it okay, from the University of Lausanne. And the second is Tina Keller Costa from the Instituto Superior Técnico in Lisbon. So Tina Keller Costa, since 2020, is a assistant professor in the Institute for Bioengineering and Biosciences, where she's also a researcher scientist in the microbial ecology and evolution group. And she's interested in both the ecological role, but also the biotechnological potential of symbiosis. And today she will tell us more about octocoral symbiosis. The stage is yours.
Thank you so much.
I cannot see you. Now I see. Yes.
Can you see my screen?
Yes.
Presentation mode. Okay.
Yes.
And you can hear me well. Perfect. Thank you so much for this nice introduction. And also for this invitation to present this work here today and to get to know your network, which is really exciting. So my name is Tina Keller Costa, and today I want to share with you about the research that I've been conducting since I started my postdoc on the microbiome of octocorals, which has become one of my favorite model hosts, and looking into community structure, function, diversity, possible metabolic interaction, and also biotechnological appliances that may arise from there. So let me jump right into just briefly say what octocorals are. So obviously corals are animals, right? They're sessile marine invertebrates. They're colonial. Their body unit is a polyp, and they're really important foundational organisms in marine ecosystems, where they increase habitat complexity and biodiversity. They are also having an important role in determining the carbon flux between benthic and pelagic zones. And as we know, they are very delicate and threatened nowadays by long-lasting disturbances and temperature rises, wherefore there is a worldwide decline of corals going on. And then when we talk about octocorals, so we have two major groups of corals, the hexacorals, which are characterized by the six-fold symmetry of their polyps. They often have this calcium carbonate exoskeleton. These are these typical reef-building corals. And then the octocorals, on the other hand, this is another major subclass of corals where we have an eight-fold symmetry of polyps, and often they lack this rigid calcium carbonate exoskeleton. And we also refer to them sometimes as soft corals. There's one particular group of octocorals that is very abundant here on our Portuguese coast, the so-called gorgonians, which are the model organisms that we study more. So they are having an internal axis made out of gorgonine. And these gorgonians, particularly the ones that we find here in our Portuguese coast, they often lack this typical algae symbiont, so the Symbiodinaceae symbiont that we know that supply a lot of carbon to the coral. So we believe that these gorgonians here, they rely mostly on prokaryotic partners, and there's a lot of heterotrophic feeding going on particles and zoo and phytoplankton. So one of the first studies that we conducted here was actually using the gorgonian coral Unicella labiata, which is the most abundant coral on our beautiful Algarve coast. And it was a very simple amplicon diversity study, asking who's there, which are the specific symbionts, but also trying to invest in cultivation and how much can we cultivate. And so in this amplicon study, it was a typical amplicon workflow, and I don't need to walk you through all of the steps, but what I would like to point out for this assessment of how much of the community we can cultivate, what we did was not only doing this traditional cultivation by picking colonies and isolating, but also then after 4 weeks of incubation at a low temperature to really allow a wide diversity of bacterial associates to flourish on those plates. We washed those plates with artificial seawater and then prepared microbial pellets from it and subjected it also to sequencing together with the natural microbiome and the seawater samples for a direct comparison. And so we have here the genus level taxonomic composition, and as you can see, In seawater, we have a very different taxonomic composition of the seawater microbiome compared to the coral microbiome. There's obviously a selection process going on. We have these typical dominant symbionts here of the Endocercomonadaceae family that is known from many corals and many studies all over the world. That this is a very typical symbiont of marine invertebrate. Unfortunately, it's a very difficult to cultivate one. There are only few phylotypes in culture until today. But we do see, if we compare this plate wash, that we do have an overlap of a number of genera like Huberia, Sphinogaptus, Chevanella, Lapenza, and so on. So, and if we translate this to how many shared versus specific phylotypes we do have here between this cultural fraction and the uncultured fraction, then it's actually quite a lot. And we can see that in this coral, we may be able to cultivate up to 62% of the bacterial phylotypes, which is really quite a lot compared to what we have just heard from soil samples, for example, where you have usually a very small percentage that is culturable, which makes this really a nice model organism. And by now we have a quite nice culture collection of about 315 isolates from these corals. They are continuously growing. Right now we are cultivating again. They are spanning 4 different phyla, 16 families, and 28 genera so far. And what are we doing with this culture collection? This is not the major topic of my talk today, but we had this project over the past 3 years where we really wanted to, called SymbioReactor, where we wanted to look into the secondary metabolite coding potential of these symbionts that we have isolated. So we have sequenced a lot of genomes, screened them for secondary metabolite encoding biosynthetic gene clusters, and also a lot of antimicrobial, antibacterial, antifungal activity screenings. And our PhD student, Sandra, she has been focusing particularly on one marine bacterium called Aquimarina that we isolate frequently from corals and also from sponges. And in her project, she has been able, in collaboration with Professor Jörg Pierle from the ETH Zurich, to isolate and elucidate the structure of a novel non-ribosomal peptide. Unfortunately, I'm not allowed yet to show the structure to you. That may be in a future talk, but I think it is a nice example how we can benefit and use from this cultivation efforts that is often cumbersome to have novel biotechnological appliances arising from that. Another example where we are going to implement this culture collection is now with our starting PhD student Mathilde, who in collaboration with Raquel Peixoto from KAUST, who is a pioneer on coral probiotics, and also in collaboration with the Lisbon Oceanarium, we will try to work towards implementation of microbiome manipulation in corals, which is very difficult compared to some terrestrial systems that you may know, because you have all these aquariums condition, etc., to take care of, and also working towards microbiome therapy for octocorals to improve the resilience of these holobiomes. But this may be more in the future. So today, what I would like to share with you are our recent studies on metagenomics, insights on the taxonomy function and dysbiosis of prokaryotic communities. And here in this particular study, we had 3 different octocoral species, Unicella verrucosa, Unicella gacella, and Leptodorgia zamatosa. We had the opportunity from one coral species to sample both healthy tissue and necrotic tissue from the same specimens, and also compare this with the surrounding environment. So we also sequenced the sediment samples and the seawater samples. And we were really trying to address this function. So which functions could serve as indicators of octocoral holobiont health, and what may be important for health, and what may they be doing. So we had these 20 metagenome samples sequenced on an Illumina platform. And then here in this multivariate analysis, you can see very nicely a separate clustering, both on the taxonomic level, so filtering out the 16S reads, and also on the functional level. We have assembled these metagenomes into context and then performed protein family annotation and cluster of autologous groups of protein annotation. And you see that the sediment and seawater samples, they form separate clusters, and this both on the taxonomic and functional level. And also in the necrotic samples here, these triangles, you see a separate clustering from the healthy samples. And what's quite interesting, particularly on the functional level, is that the necrotic samples and their functional profile of the microbiome of the necrotic coral samples starts to resemble much more the functional profile of the microbiome of the seawater samples. And looking a bit more into the family level taxonomic composition of this data set, so obviously you have a very different microbiome associated to sediment and seawater than you have in the corals. But if you look into the coral samples, you really find again this Endocercomonas group that I also showed you in this previous amplicon dependent study. Here we have an amplicon independent study. They are really abundant in the different coral species. And you see a large decline when the coral tissue gets diseased in all of these samples. And what is also interesting here to note is if you compare the two coral species from the same genus, so Unicella gazella and Unicella verrucosa, the healthy profiles are actually quite similar. And then if you move to the species that is from a different genus, the coral species from a different genus, you see that the dominant members there are still shared, but the community profile changes. So there is also indication of phylosymbiosis here in relation to the octocoral host. Looking then more into the functional signatures of the microbiome of healthy octocoral tissue. So which are the characteristics of the microbiome of the healthy octocoral holobiont? Well, we see a lot of features enriched in DNA repair and genome stability, such as exonucleases, for example. We have a plethora of features involved in antiviral defense, a lot of CRISPR-Cas proteins, CRISPR-Cas endonucleases, restriction endonucleases, and also negative regulators of phage lambda lysogenization. So this bacterial community is well equipped to counteract antiviral attacks. And actually, if you look at the viral load in the samples, it correlates very nicely to what we see. We have a much higher viral load in the seawater samples compared to the healthy coral samples, and also a higher load in the necrotic coral tissue compared to the healthy coral tissue. So there's a very nice correspondence here. We also have features involved in host symbiont recognition, such as eukaryotic-like repeat proteins, and particularly eukarine repeats, significantly enriched in the healthy holobiont. And these are features that are eukarine repeats are involved in protein-protein interactions, and they are found across the 3 domains of life. They are first known from eukaryotes and have been implicated in host-microbe horizontal gene transfer. And what happens is that in host-associated marine invertebrates, we often see a higher abundance of these genes in comparison to the free-living microbes or free-living bacteria. And there has been experimental evidence from the group of Thurston Thomas in Australia that if you overexpress this eukaryotes on the membrane of an E. coli, they can evade phagocytosis von Amoebae. So this is another factor that we believe stabilizes this symbiosis by evading phagocytosis and being involved in host symbiont recognition. We have also other features involved in stress response and carbohydrate transport and iron storage and ion transport, inorganic ion transport, particularly enriched in the microbiome of healthy coral samples. And then if we move to the microbiome of necrotic coral tissue, the features that we see increasing there are a lot of features involved on pathogenicity, virulence, and host invasion. So an increased abundance of type 6 secretion system, virulence factors in invasion proteins. We also have increased number of genes of nitric oxide reductase and arginase. These are probably involved in host immune system attack. So corals are known to react to a pathogen invasion or to a stress with a nitric oxide reproduction response, or a nitric oxide burst. And then on the other side, pathogens may have mechanisms to counteract on this nitric oxide response by using nitric oxide reductase or the arginase, which drains the common substrate from the nitric oxide synthase of the corals. We further see features involved in chemotaxis and chemical quorum sensing and chemical communication of this invading microbiome. So N-acetyl homoserine lactose synthase encoding genes are also increasing, and several features involved in fatty acid utilization, sugar and amino acid degradation, all together suggesting actually a feasting of this pathobiome on the dying or decaying coral tissue. So with this study, we had an understanding of what may be feature important in stabilizing symbiont-host relationships in the octocoralline holobiome, such as the antiviral defense system, such as the Ankai ring repeats. But we still wanted to go a bit further, addressing this question, really who does what. So we also assembled metagenomes. So we also prepared this metagenome assembled metagenomes from this data set and really deeply explored those metagenome assembled genomes. And this is still work under review, but the preprint is already available. So I'm happy to share this with you today. So we have retrieved 66 mags from this data set, 65 of them bacteria, 1 archaea, all with quite good completeness scores of 80% or higher and low contamination scores, which for coral samples isn't always so easy. And these 66 mags are divided into 30 different species. And what is quite interesting is that the 30 species are really depending or correlating with the microhabitat that they come from. So we have very little overlap from what comes from the seawater to the necrotic tissue and to the healthy tissue. And I would like to call your attention here on these Gorgonimonas, as we call them now, mags. So we have retrieved 11 Endocercomonadaceae mags from from nearly all the healthy octocoral samples. After some deep phylogenomics analysis of these 11 Endocercomonas mags, comparing all Endocercomonas genomes that we have in the database, we could conclude that these mags really belong to 2 novel species of a novel genus that is unique of temperate octocorals, which we now propose this name here, Candidatus Gorgonimonas. And then I have been comparing these 11 mags against all the other mags in the database, against all the other mags in our data set, and to really infer using KEGG and COG annotations, what are the characteristics of these genomes and what may these symbionts be doing. So what's Gorgonimonas doing? So I came up with this schematic drawing here of a predicted metabolic features. So we do indeed see again this antiviral defense system, the Ankai ring repeats that we have been seeing enriched in the healthy coral samples. Now we can attribute them to these symbionts. We also see a lot of features involved in TFP pilus assemble that may indicate adherence and aggregation. A lot of features involved in inorganic ion transport again, particularly also iron 2 acquisition, which may indicate that they may also be amenable to suboxic conditions. And there are other metabolic features that we see that may actually indicate that these symbionts may also pursue a facultative anaerobic lifestyle. And one feature that I would like to point out to you that is particularly interesting, all these 11 mags, they harbor consistently chitinase encoding genes and chitin binding proteins, and chitinases are enzymes that degrade the large chitin polymer, which is the most abundant sugar in our oceans. So it has a big implication in carbon and nitrogen cycle in our oceans. So all these 11 mags have a complete chitinase gene with a signal peptide present, indicating that it can also be excreted from the cell. And when then taking this into a perspective from this community of mags that we have from the healthy coral symbiont, what is quite interesting is, so these Gorgonimonas, these Endocercomonas symbionts, they have all these endokitinases to degrade the large polymer, and then we have others here, like these Metamycoplasmataceae and the Thioglobaceae, other groups of symbionts that may, for example, be able to consume the derived degradation products. So we propose that this might be a mechanism of cross-feeding going on, and Thioglobaceae also promote degradation of the polymer by having chitin deacetylases. So this is one way we believe there may be metabolic interaction in the holobiont, and which may also contribute that in some of these symbionts you have this very reduced genome size. And there are many other features that are very interesting to discuss. But out of time, I don't think I will go any further here into this graph. I just would like to stay a little bit more on this kiting story, because we also had another study recently where we looked into differential chitin degradation and utilization features across free living and host-associated marine microbiomes, not only using this octocoral data set, but also using sponge metagenomic data. And it really turns out that if you look in the octocoral data set in terms of the endochitinase encoding genes, the number of genes level up to what you also have in the surrounding environment, being a further indication that chitin degradation and chitin processing may actually be an important mechanism of nutrient turnover in this holobiont. And I believe it makes a lot of sense because these octocorals, after all, they are suspension feeding on large quantities of chitin-rich particles, such as phytoplankton and zooplankton, that also often have chitin in their cell walls. So having a microbiome that is able to contribute in chitin turnover would be a beneficial thing. So now you may say, well, this is all metagenomics evidence only, and that is true. And we are now working on a new project that I just got funded, where we will try to use synthetic biology, because these Gorgonimonas, Candidatus gorgonimonas, so far is not culturable, or we are not able to retrieve it in culture, but we are also keep working on that. But with this project, the idea is to use synthetic biology and heterologous expression to harness some of these chitinases that we see in these symbionts and to characterize their function better. And this is work that is now starting. And with this, I'm really already at the end of my talk. I would like to thank all my colleagues and co-authors for their contributions, particularly Rodrigo, our wonderful MicroEco-Evo team, all the fungi agencies involved here. And of course, thank you very much for listening. And if you have questions, I'll be happy to answer.
Thanks a lot, Tina. Is there any question from the audience? If so, please raise your hand or write it in the chat. Otherwise, I could start by asking one question or maybe two. So the first one, it's very naive because I don't know the biology of these octocorals, but when you sample the microbiota, does this microbiota live in some sort of pocket or like pores we have in our skin, or do this structure exist? And if so, are these structures maintained in a necrotic tissue or not?
Yeah, that is an excellent question, and that's really something that is still difficult to answer. So there has been one study from the lab from Christian Wolz in I think in 2017, where they have so people are trying to locate these symbionts down, where in the coral tissue they are. And this one study was locating the Endosarcomonas symbiont down really to sit deep inside the endoderm and also in these polyps that they have, and was able to show that they have formed some dense aggregates there. But apart from this, we really know relatively little. I think now there are more studies trying to separate skeleton from the tissue and from the mucus. So we know that the mucus associated microbiome is often this different, for example, it's more this external part. But for these gorgonians in specific, it is really not easy to separate the different compartments because you have relatively little tissue to work with from the beginning, and then to prepare good metagenomic samples from this. So I had to actually scrape a lot of tissue to get to it. But it's definitely something that we need to explore in more detail. Yeah.
Okay, I was asking because I was wondering whether the differences that you see in the necrotic tissue would or could be justified by a sort of increased immigration from the seawater.
Definitely, yeah, definitely. I think that's what we are already seeing. And on the functional level, if you look at the overall functional profiles, you do have already this closer functional profiles from the seawater. So, and you also see increase of certain taxa, for example, Rhodobacteriaceae taxa in the necrotic tissue, that we also see highly abundant than in the seawater. I didn't show this today, but yes, that's definitely going on. That there is then this probably already the secondary colonization also, right? Yes.
Thank you.